Welcome back. We are looking at the transfer coefficient approach. We are looking at it in the context of oxygen supply to bioreactors. Now we are in the middle of a generalized formulation, a reasonably gentle formulation for this approach itself. We said that we are trying to represent the transport across phases through appropriate definitions or appropriate quantities. The essential difficulty being, if you want to work only with concentrations and not with chemical potentials, then concentrations you can do algebraic operations over concentrations that are in the same phase. For example, we could add, subtract YAG, YAI, and so on, or XAI, XAL, and so on. But you cannot add, subtract YAG, XAL, and things like that for obvious reasons, because the base aspects are different and so on and so forth. So you cannot represent the difference as YAG minus XAL directly and expect it to hold. Okay. To overcome that difficulty, we are looking at a conceptual quantity. We said that we will define a quantity in the gas phase that will be in equilibrium with the bulk concentration of the liquid phase, and thereby we could do the algebraic manipulations. So that is what is given here. Na, the flux of A, is a product of an overall transfer coefficient K, capital Ky, times YAG minus YA star. YA star is the conceptual quantity, it's not a real quantity. This a real quantity in a certain sense, but it is not real in the way we are describing the process. That's all. The YA star is the concentration in the gas phase that will be in equilibrium with the bulk liquid phase concentration, and thereby we have transformed the bulk liquid phase concentration into an equivalent gas phase concentration that we can do algebraic operations on. That is for this. So Na equals capital Ky times YAG minus YA star. Similarly, we could do the same thing for a liquid side aspect. The flux of A equals a capital Kx, an overall transfer coefficient in terms of the liquid side X, times XA star minus XAL. XAL is the bulk concentration here. XA star represents the concentration in the liquid phase that will be in equilibrium with YAG, and therefore that provides the needed driving force. So by this process, we have expressed the flux in terms of a difference between concentration in one phase and an equivalent concentration in the other phase, and also in terms of an overall coefficient. At steady state, there should be no accumulation at the interface, because the properties of interest, concentration, does not change with time. If there is accumulation, the concentration of the species will change with time. Therefore, the flux of A reaching this interface must equal the flux of A leaving the interface. Okay? It's a powerful view which we have already used. Let's use it again. In a totally different context, we used it. Recall where we used it. Here, at steady state, there should be no accumulation at the interface, and therefore the flux of A reaching the interface must equal the flux of A leaving the interface. So what is the flux of A reaching the interface in terms of individual mass transfer coefficients? Lowercase ky, YAG minus YAI is the rate at which oxygen reaches the interface. Kx times XAI minus XAL is the rate at which it leaves the interface, and both those must be equal, and both those were equal to Na from our expression anyway earlier. Okay. So we are equating this at steady state. 6.2.1-12 equation number. The flux Na can be written in terms of the overall mass transfer coefficients. These are the individual side mass transfer coefficients. In terms of the overall mass transfer coefficients, the two expressions need to be the same because they represent the same flux, and therefore Na equals capital Ky, the overall mass transfer coefficient, times YAG minus a concentration that is equivalent to the bulk liquid phase concentration, Ya star, that's in equilibrium with the bulk liquid phase concentration, equals Kx times Xa star. Xa star is the liquid side concentration, which is in equilibrium with the bulk gas concentration. Therefore, it represents YAG minus XAL. Okay. So this is the same flux NA. This is written in terms of individual mass transfer coefficients. This is written in terms of the overall mass transfer coefficient. Equation 6.2.1-13. Now recall that the equilibrium curve is the relationship between concentrations of species in a liquid and gas phases at equilibrium. Y versus X, and the relationship is something like this. This is at equilibrium. That's a typical equilibrium relationship or equilibrium curve. If the equilibrium curve is linear or can be considered to be piecewise linear in the region of interest, YAI, the interfacial concentration, remember, at the interface, there is equilibrium between the gas and liquid phases, instantaneous equilibrium. So YAI equals the constant M times XAI. Okay. A linear constant we have taken here. In other words, we have taken a region of relevance in the equilibrium curve, which could be this curve kind of a thing. We have taken a region small enough so that you can neglect the curvature and take it to be a linear relationship. Equation 6.2.1-14. Therefore, YA star can be written as M times XA. Right? It's equilibrium concentration. So, YA star is the concentration that is in equilibrium with the bulk liquid phase concentration. YA star equals M times XA. Equation 6.2.1-15. From equation 6.2.1-12, which is the side, which is the flux written in terms of individual mass transfer coefficients, YAG minus YAI is nothing but NA times KY. We just taken KY to the denominator on the other side. 6.2.1-16. Therefore, what is YAG? M times XA star, right? That we saw from this equation here. YAG is M times XA star minus YAI, the interface concentration at equilibrium, XAI, M times XA star minus XAI equals NA by KY. So XA star minus XAI is, you take M to the other side, NA by MKY, equation 6.2.1-23. And also from equation 6.2.1-12, the other side of it, we get XAI minus XAL equals NA by KX. Just to fine tune things, we are taking these two parts of this equation here, XAI minus XAL equals NA by KX. Okay. 6.2.1-24. And adding 6.2.1-22 to 6.2.1-23, so this has to be 24. Yeah, I'm adding this to this. Say star minus, yeah. This has to be 24 to 23, this to this. XA star minus XAL, you add these two together. XAI minus XAI are going to cancel out. So XA star minus XAL equals NA common out 1 by MKY plus 1 by KX. We would call this equation 6.2.1-25. Let me correct this here. It's 23 to 24.3. Yeah, we are fine in terms of those numbers there also. Okay. Also from equation 6.2.1-13, which happens to be the other one, right? This is there. This is in terms of the overall mass transfer coefficient. See this? XA star minus XAL equals NA divided by capital KX, the overall mass transfer coefficient, 6.2.1-26. And if we equate the right hand sides, since the left hand sides are the same of equation this and the previous equation, XA star minus XAL, XA star minus XAL. Okay, so if we equate those, we are going and cancel out NAs, of course. We're going to get 1 by capital KX equals 1 by M times KY plus 1 by KX. Equation 6.2.1-27. If M is large, okay, what is M? M is the relationship between the gas phase concentration, mole fraction, and the liquid phase mole fraction. Okay. In other words, if it is a sparingly soluble gas, sparingly soluble species, then at equilibrium the gas phase concentration will be much higher than the liquid phase concentration. Okay, so that is the situation here. Let us say a sparingly soluble gas. Oxygen is a sparingly soluble gas. You would recall that the thumb rule value is about 8 ppm under normal conditions of temperature and pressure. Is the oxygen concentration in the liquid, right, at equilibrium. So 8 ppm is small. So M is indeed large. If M is large in comparison with the other things, this becomes small in comparison with this, and therefore this drops out. Therefore, 1 by capital Kx is approximately equal to 1 by lowercase kx. In other words, the overall resistance, this is some sort of a conductance, right? So the inverse of the conductance is the resistance. The overall resistance here given by the left hand side is approximately equal to the resistance in the liquid phase. The resistance lies entirely in the liquid phase. This implies that the mass transfer flux is determined by the gas side mass transfer flux. In terms of the overall mass transfer coefficient, flux is nothing but the moles transferred per time per unit area perpendicular to the direction of transfer. So that is flux in our formulation is capital Kx times Xa star minus Xal. Okay. So since this is the equilibrium concentration, this can be calculated, this can be measured, and as long as we find, there are ways of finding this, so this can be handled. Okay, you will have a measure of this by this kind of formulation. So mole transfer rate is moles transferred per time, which is what is more important for us. It will be flux times the area. The transfer rate is the flux times the area. So Kx into Xa star minus Xal times A. A is the interfacial area because it transferred across the interface between the gas bubble and the liquid, or gas bubbles and the liquid, considering a single bubble. So moles transferred per unit volume per time, because we like to normalize things in terms of volume, is we divide this expression here by the volume of the bioreactor, by V. So Kx A by V, Xa star minus Xal. And this A by V is typically represented as the lowercase a, the surface area per unit volume, the lowercase a. So moles transferred per unit volume per time becomes overall transfer coefficient Kx times A, Kxa times Xa star minus Xal. A is interfacial area per unit volume, equation 6.2.1-28. And again, the interfacial area cannot be measured easily. Like the interfacial area, remember, it's the area of the bubbles, the interface between the bubbles and the liquid. That's not very easy to measure. Therefore, by an appropriate means, Kxa is measured as a single variable. And that's the reason why in bioreactors, the volumetric oxygen transfer coefficient, right, moles transferred per volume, the coefficient for that, moles transferred per volume per time, that is this times the concentration difference. KXA for oxygen, is often referred to as KLa. And of course, this is also written in terms of concentration difference rather than the mole fraction difference. That doesn't matter because total concentration times the mole fraction is the concentration of A, right? So it doesn't really matter. Of course, the KXA is going to be different from the KLa in terms of the concentration and the mole fraction, but the form of this remains the same. So in bioreactors, the volumetric oxygen transfer coefficient KXA is often referred to as KLa. KLa is one of the important parameters that is determined before each bioreactor operation that gives us an idea as to how much oxygen can be transferred per volume to the cells in culture. So that's a very important parameter to know. We need to know whether we are able to supply the oxygen that is required by the microbes for them to grow and produce the product of it. So that's a measure here. It provides a measure of the volumetric oxygen transfer capacity in the bioreactor. I think we will stop here. You had things to soak in, right? You need to mull over them and then become comfortable with them. In the next class, next lecture, I'm going to tell you about how to estimate this KLa, which is a very important thing for biological engineers, something that is commonly done, whether you whether you run a bioreactor in the industry, whether you run a bioreactor in the lab that looks at appropriate aspects. You certainly need to measure KLa pretty much before every run. Okay, let's meet in the next class and take things forward. See you.